Hello everyone. In this research, we looked at the effect of jaw clenching on persistent inward currents of spinal motor neurons. When we clench our teeth, we can produce greater muscle forces, but we don't really know why. An underlying mechanism could be an enhancement of the activity of persistent inward currents, or PICs, of spinal motor neurons. PICs are activated by special channels which induce a persistent or ongoing flow of positively charged ions into the cell, which facilitates motor neuron firing. We also know that these peaks are enhanced by the release of serotonin and noradrenaline onto these nerve cells. Given that we release serotonin when we contract our muscles, we wanted to see if clenching our teeth would have an effect on peak strength in plantar flexor motor neurons. So we asked our 21 participants to perform isometric ramp contractions of plantar flexors during control and jaw clenching trials, where participants clenched their teeth on a mouth guard as hard as they could. High-density surface EMG of the gastrocnemius medialis was recorded, and the EMG signals were decomposed into motor unit discharge timings and tracked between conditions. 2 ramps from each condition were considered for further analysis, and the instantaneous firing frequencies were smoothed by a 5th or 4th order polynomial. We used the delta F technique, which uses pairs of motor units to estimate peak strength. A lower threshold motor unit, the control unit, is used as a surrogate of net excitatory synaptic input, allowing the quantification of the level of this input at recruitment and de-recruitment of a higher threshold motor unit, the test unit. The difference of this estimated input between recruitment and de-recruitment is our delta F value, and an estimation of the contribution of peaks to motor neuron firing of our test unit. Suitable pairs were found in 18 out of 21 participants. 130 test units were considered for the delta F analysis, and there was no significant change of the delta F values from control to jaw clenching. These findings do not support an effect of jaw clenching on peak strength of the gastrocnemius medialis during a low-intensity contraction. Possible explanations for the absence of an effect include a ceiling effect. Performing a ramp contraction already increases descending serotonergic drive, which might decrease the likelihood of further neuromodulation, or a regionalization effect, as jaw clenching might influence other motor neuron pools, such as soleus, but not the gastrocmedialis. These findings are part of a bigger project, and I would like to thank all the collaborators and thank you for listening.